Hello, everyone, and welcome to today's webinar titled Operation Moonshot, Rapid Translation of a SARS CoV2 LC-MS test from research to routine use. This webinar is part of the Clinical Diagnostics and Research event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Water Corporation. For more information about Waters Corporation, please go to waters.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our new live chat feature during the presentation. You can find this live chat feature located at the right of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of this presentation. You will also notice that the speaker has provided a survey located at the right of the abstract. This survey will be available during both live sessions and on the on-demand period provided for you. Now, if you have any trouble seeing or hearing this presentation, just click on that Help Desk button located at the bottom of your screen where the navigation bar is or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Rachel Carling, the Scientific Director at Viapath, Guy's and St. Thomas NHSFT. For a complete biography of our speaker, please visit the Biography tab at the top left of your screen. Dr. Carling, welcome. You may now begin your presentation.
Thank you, Lance, and thank you for inviting me to talk today. My day job is actually metabolic biochemistry and newborn screening. So virtually all the tests we do in my lab involve chromatography or mass spec in some way, shape or form. So when in the middle of the pandemic last year, the Department of Health were looking for clinical labs with mass spec experience, we fitted the bill nicely and we were invited to participate in the Moonshot project. This was a project set up in summer 2020 when there was concern over the UK's capacity for COVID testing. And the aim of the Moonshot project was to identify alternative technologies to PCR for detecting COVID. So my talk is kind of going to be in two halves. In the first half, I'm going to talk about the Moonshot project itself, and then the second half will focus on some of the developments which have occurred since then. So I think it's probably just worth reminding everyone what the definition of a moonshot is, because I think it helps to remind ourselves of the context in which this project originated and just how ambitious it really was. So a moonshot is an ambitious, exploratory or groundbreaking project undertaken without any expectation of near-term profitability or benefit and without a full investigation of the potential risks and benefits. So the Moonshot project was set up by the Department of Health in summer last year, and it was a unique collaboration between academia, industry and the NHS. The academics were tasked with developing a mass spec method for SARS CoV2 testing, and the NHS clinical labs were then tasked with operationalising the method, undertaking a formal method comparison and validating the method to ISO 15189 standards. And of course, the entire process was supported by our industry partners. So the academics involved with Moonshot were Perdita Barron from Manchester, Paul Skip from Southampton, Don Jones from Leicester, and Kevin Mills from London. And all of these people are amazing scientists, and they worked together to come up with this method in a very short space of time. They've been given set performance criteria by the Department of Health that they had to meet. So the method needed to detect between 2 and 5 target peptides from either the nucleocapsid or the spike protein. It also needed to have 95% sensitivity and specificity at a CT of around 25 to 30, and it needed to be robust and lend itself to scale up. So all in all, I think you will probably agree that the specification was in keeping with the definition of a moonshot. But these guys are really good, and they came up with version 1 of the method by December last year. And it quickly became evident, though, that with just the SPE in place, we didn't have the required sensitivity. We were also struggling to get hold of positive samples to play with. And given that we were in the middle of a global pandemic, that was really quite ironic. However, they were undeterred, and by January this year, they'd introduced ciscapper into the workflow. So ciscapper stands for Stable Isotope Standard Capture Antipeptide Antibody. And the ciscapper really boosted the sensitivity of the method quite significantly. And so it was ready to be translated into the clinical laboratories. So I'll just give you a brief overview of the method that the academic labs developed. So the swab was deactivated at the point of collection by collecting the sample into an ethanol buffer. Now, this was really quite an elegant solution because none of the academic or clinical labs involved in the Moonshot process had access to Category 3 facilities to deactivate the samples. So this was quite important. The method required both the swab and the ethanol buffer it was collected in to be processed. So we refer to the ethanol buffer as a swab juice, and we needed to process the swab as well because the ethanol fixed about half of the viral material onto the swab. So the method followed a fairly standard peptide workflow. The protein was precipitated and denatured with Rapigest prior to undergoing a triptych digestion to release the constituent peptides. And there were 3 target peptides in the method: AYN, ADE, and DGI. And these peptides were isolated using ciscapper technology prior to detection by mass spec. So I'll just talk a little bit more about ciscapper in case you're not familiar with how it works. So as I said, it stands for Stable Isotope Standard and Capture by Antipeptide Antibody, and it's a really clever technology. The antibodies against the target peptides are coated onto magnetic particles, which can then be incubated with a digested sample. And the antibodies capture the target peptides, and after the particles have been washed, the target peptides can be eluted and injected into the mass spec. And basically, the ciscapper step replaced the SPE in the original method and really improved the sensitivity. And then what this slide is just trying to illustrate is how the process actually works in practice. So the brown solution that you can see in plate 1 in the picture is the magnetic particles once they've been added to the digested swab samples. The plate is then incubated at room temperature for an hour and then placed onto the magnetic array, which is shown here in picture 2. And the magnets sit in between any 4 wells on the plate and they pull the beads to one side. And hopefully in picture 3, you can just see the pellet of beads that's formed at the corner of each well. And once this has happened, you can go in with a pipette, and obviously you can use a multi-channel pipette, and you can remove the solution. And then the process is repeated a couple more times before the peptides are eluted from the beads. So as one of the clinical labs involved in this project, I think it is fair to say that in many respects, the academic labs have done all the hard work for us. However, I think it's probably worth just saying a little bit about the analytical challenge that this project presented for the clinical chemistry labs. So at the moment, virtually all clinical mass spec work in the UK is small molecule work. We offer quantitative assays, which we hope are accurate, precise and selective. And we measure the concentration of an analyte in a fixed amount of biological material, be that plasma, urine or a dry blood spot. Most of the analytes that we measure are present at around 0.1 to 100 micromolar concentration. And although most LC-MS methods are limited somewhat by the lack of automation and pre-analytics, they are typically considered to be the gold standard method of analysis. So proteomics was an entirely new field for us. We normally precipitate the protein and keep the supernatant, and all of a sudden we were doing the reverse. And to be honest, it did require a little bit of getting your head round. So we were measuring peptides, but because we were working with swab samples, we had no idea how much biological material we had started with. So we couldn't report an actual concentration. All we could really do was determine the amount of material that we'd injected onto the column. And this, again, was an entirely unfamiliar concept to me. And of course, the concentration range that we were trying to measure at literally blew my mind. We were looking at concentrations that were 6 to 7 orders of magnitude lower than anything I'd ever known a triple quad do. But I think for me, one of the best things about this project was that in December last year, we got mass specs with this kind of capability installed into my lab at St Thomas's. And here they are. So these were my 2 shiny new Waters Zevo TQXSs. I named them, obviously, and I called them Frank and Leslie. And this is after Frank Pick and Leslie Green of London Underground fame. So Frank was the chief executive of London Passenger Transport Board, and Leslie was the architect who was responsible for the characteristic oxblood red tube stations and the really lovely platform tiling patterns, which some of you might be familiar with. But Frank and Leslie were creative, strategic and visionary. And I think that was really just like the Moonshot project was, so it seemed like a good name for them. So thanks to our industry partners, we had the kit, and thanks to the academic labs, we had a method. So our job as clinical labs was then to sort of optimize the method to make it work in the routine clinical environment and do a method comparison with PCR and validate the method to ISO 15189 standards. So we took a fairly evidence-based approach to this, and we did make some minor tweaks to the method, but primarily we focused on identifying a calibration strategy, introducing quality control, and working out how to determine whether a result was positive or negative. Once we'd done this, we did a fairly straightforward method validation and then the method comparison with the PCR samples, which of course PCR is considered to be the gold standard method of analysis for COVID testing. And it's probably also just worth mentioning that the other thing we did was to firmly badge the method as qualitative. And although from the UCAS perspective, it would be classed as semi-quantitative because we are generating numerical results to assist with the classification, in my opinion, the method is essentially qualitative. So I'll just take you through the highlights of the method validation now. So we dropped the DGI peptide from the method quite early on because we found that there were significant issues with carryover, and it also lacked the sensitivity of the other two peptides. But as a learning point, it is always useful to include a number of target peptides when you're starting to develop a method, so you can work out which ones perform best as you go along. So luckily, the other two peptides, AYN and ADE, performed well. We found that they had acceptable sensitivity and precision and negligible carryover. We also found that the method was selective. There was no cross-reactivity from influenza A, influenza B, or RSV. And until the Delta variant came along, the target peptides were unaffected by the mutations. So, however, the Delta variant did result in a mutation on the ADE peptide, and this slide is just trying to describe this visually for you. So these, highlighted in green here, you can see the three target peptides that we've been looking at, the DGI, the AYN, and the ADE. But the Delta variant resulted in 8 different point mutations, and these are highlighted in the peptide sequence in red. And the D377Y mutation was on the ADE target peptide, and that's circled. And what happened to it was that the ADE became AYE in the Delta. So this mutation reduced the affinity of the ciscapper for the ADE peptide, or should I say for the AYE peptide, but luckily only by approximately 20%. So by adding in the MRMs for AYE into the mass spec method, we were still able to detect the Delta variant, which was a real bonus. So we did a method comparison against the Thermo Fisher Takehome PCR assay, and we compared swabs from 396 subjects, which were collected as part of the Falcon study. And these samples were collected from people in hospital by healthcare professionals, but also by people screening themselves at home as part of the population testing programme. We had to exclude 35 samples due to either poor sample quality or an inconclusive PCR result, which left us with 361 in the final method comparison. And we found that the mass spec method had a sensitivity of 92.4% and a specificity of 97% compared with PCR. And that gave a positive predictive value of 96.9%. And Cohen's kappa demonstrated that there was excellent agreement between the methods, which was really good. I think it's also worth pointing out here that the PCR and mass spec results were coming from different swab samples. So the efficacy of the swab sampling process will also be impacting on the comparison between the two. And to try to put that in a little bit of context, a comparison of just over 500 paired swab samples, which were both measured by PCR, the same PCR method, but in different labs, that gave an error rate of 3%. And because there is a lot of discussion and perhaps a lack of clarity around what a CT value above 32 actually means in terms of someone's infectiousness, we did split out those samples with a CT of greater than 32 and just have a look at what that did to the sensitivity. And as you would expect, it did improve it significantly. As to whether or not that's an acceptable thing to do, I think I would leave that to our virologists to comment on. So I think in terms of the original ask, the Moonshot project was really successful. An LC-MS method had been developed very quickly and translated into the clinical lab where it had been validated, and both the analytical performance and the comparison with PCR were acceptable. However, the method did also have some limitations, which meant it wasn't really that suitable for the routine clinical laboratory. I think for me, the main one was the bespoke sample collection tube. So although it was ingenious in its own way, it wasn't logistically feasible. And in fact, it made access to patient samples incredibly difficult. And the table shown here on the slide just gives you an idea of just how many samples were received in an incorrect volume of ethanol, which is a reflection of the tubes being prepared manually and not through a kind of commercial controlled process. And also, because both the swab and the swab juice had to be processed, the method was really a one-shot method, which obviously isn't ideal in a routine lab because you can never go back. You've just got one chance to get it right. And again, processing the swab without automation was quite tricky. And perhaps more importantly, there was a real risk of cross-contamination from doing that. And then on top of that, the method was incredibly labour-intensive and time-consuming. So the mass spec aspect was really pretty fast, but the rate-limiting step was the manual sample preparation, which really did mean that if you were doing this manually without any automation, you could really only prep 20 samples in a day. So I think for me, for the method to be suitable for use in a routine clinical lab, the first thing that needed to change was the sample type. I mean, the world and his wife are collecting swabs for COVID into BTM, and we needed the method to work on BTM. We also needed the process to be automated, partly for ease and simplicity, but also so the throughput was increased. And I think ideally, we needed a post-analytical data processing solution because the existing process required review of the numerical data and visual inspection, quite careful visual inspection of every single chromatogram. So I think really this is where our industry partners came into their own. So my lab had been partnered with Waters, and we've been having some ongoing discussions about how the method might work with BTM samples. But progress in actually doing something was slow, partly because of the difficulty of getting hold of reagents. So the ciscapper was like gold dust in January, February time this year. But also getting hold of positive samples was quite difficult, and this did slow down our progress. But Waters came up with a reagent kit, which was designed to work with BTM samples, which solved two of our problems straight away. And then they also loaned us a benchtop robot, the Andrews Alliance, to help with the sample preparation, and this had such a positive impact. We also sent our swab data to them, and Hans Vissers has been busy working on an algorithm to try and classify the samples. And I was really pleased a couple of months ago when he came back and had independently verified the classifications that we'd done manually during the validation. Here's a nice picture of the SARS CoV2 reagent kit from Waters. So the kit method is really similar to the ethanol-based method I've just been describing to you, but it does have some important differences. So this kit has 3 target peptides, the AYN and the ADE that were described previously, but it also has another peptide, NPA, which is also derived from the nucleocapsid protein. All the reagents, including the ciscapper, are provided in the kit. The LC method is a little bit shorter, it's 2 minutes, and that's because the DGI peptide is no longer included. And most importantly for me, the method is intended to be used with a swab collected into BTM. So as I'm sure you can imagine, I was really keen to get my hands on some kits and try and do some validation work. So this meant I needed to find some patient samples. And luckily, with the help of some colleagues in the emergency department, I was able to obtain ethics to collect samples at St Thomas's. So our inclusion criteria was very straightforward. Any adult presenting to the emergency department between June and August this year with symptoms consistent with COVID and who consented to provide us with a second swab. So that swab sample was sent to our virology department, and they took responsibility for anonymising the samples, deactivating them, and preparing 2 aliquots. They kept 1 aliquot for PCR, and then they sent 1 aliquot to us for mass spec. So, of course, a major benefit of this comparison was that the PCR would have been done on the exact same sample as the mass spec. So that would just rule out any of those potential differences due to the efficacy of the sampling process. And by the beginning of September, we had just over 300 samples to validate the kit on. So obviously, again, we did a method comparison with PCR. This time we used the Diagnostics Method. That's a 2-step PCR assay. And just to confuse things a little bit, this assay didn't report in CT values. It used the takeoff value. So a really strong positive will amplify sufficiently in the first step of the process. And if that's immediately detectable in the second step, so that the takeoff value would effectively then be 0. So we used the Andrews Alliance for the sample prep. So this is the benchtop robot, which Waters lent us. It's a user programmable robot, and it can do a variety of things. So it can pipette, it can mix, and it can shake. And it also has a magnetic array on it, so it can do the ciscapper steps too. And basically, the Andrews did all the hard work for us. we just had to plate out the BTM samples and then transfer the plate onto the Andrews and let it do the rest. And I've just got a short clip, which I'll just see if I can play for you very quickly now. So it's just the Andrews done cutting out some of the ciscapper. So this slide is just an attempt to illustrate to you what a difference the robot made. So the preparation of the ethanol samples was entirely manual, and the rate limiting step here was really the number of ciscapper washes, because the 3 ciscapper wash steps have to be completed within 15 minutes, and that just effectively limited the number of samples that you could process in one go. And we found with the manual process that was about 24. The process was also incredibly time consuming, and it took 7 hours, and 5 hours of that was actually touch time. So by using the Andrews to prepare the ethanol samples, it meant that we could do 48 samples in one go. Again, the rate limiting step was being able to complete the 3 ciscapper wash steps in 15 minutes. And even the Andrews could only do this for 48 samples at a time. So that was the rate limiting step. It did speed the process up though. So the total time to prep 48 samples was 5¼ hours, and with a contact time of about 2¼ hours. However, Waters were able to show that the BTM method only required 2 wash steps of the ciscapper. So that meant that the Andrews could prep 96 samples in a time, which was a real game changer. So the total time was still about 5¼ hours, but the contact time was reduced to 1 hour 30 minutes. And 1 hour of that was me plating out the samples ready to go onto the Andrews. So I'm just going to walk you through the highlights of the validation that we did with the Waters kit now. So obviously we looked at linearity, and this slide shows the calibration curves for each peptide. So these calibrations are based on synthetic peptides, not on NCAP. And the peptides have been taken through the sample preparation process, even though technically they didn't need to be, just for completeness and to ensure that they were, I guess, matrix matched. We did. Have to be honest, going forward, I don't think there's any merit in doing this. And I think in future, I would just run them as offline cals. And it's also probably worth just clarifying that the AYE peptide has been calibrated against the ADE. So the table here summarizes the R² values and the back calculated concentrations for the calibrations over 5 days. And I think you can, or I hope you can see that the AYN and the ADE perform quite well, but the MPA a little bit less so. And we found that the MPA wasn't particularly stable, and it was also considerably less sensitive than the other 2 peptides. So the cal curve for NPA shown here is actually at a concentration 10 times higher than that of the other 2 peptides. In terms of imprecision, we made some 3 pooled QC samples that were made up from patient swabs collected into BTM. So we had a negative pool, a low positive pool, and high positive pool. Unfortunately, because of issues with access to samples, we were only able to obtain 1 QC for ADE, and that was a weak positive pool. We found the assay was reasonably precise. The interbatch imprecision was around 10% in the high pool and around 20% in the low positive pool. So obviously, if you're used to quantitative assays, that perhaps doesn't sound like brilliant performance, but I just think bearing in mind the concentrations that we're looking at here and the fact that this is really a qualitative assay, I think that's reasonable performance, and I think we can be confident in that. We also found that the assay was selective. We analysed 18 patient samples which were positive for one of the 4 other seasonal coronaviruses and found no cross-reactivity with COVID, so that was good. And we also looked at spiked recombinant NCAP, which with increasing concentrations of influenza A, influenza B, and RSV, and again, there was no cross-reactivity at all. So I'm just going to show you some example chromatograms so you have an idea of the kind of data that we were getting from this method. So these are the extracted ion current chromatograms from a negative sample. Each peptide has a quantifier and at least 1 qualifier ion, and of course there's a stable isotope standard included as well. So on the top here we've got AYN, sorry. In the middle we've got ADE and AYE, and NPAs at the bottom. And for each peptide, you can see here, just glancing at it, that only the stable isotopes are present. So this was a true negative. The next one we've got an example of is a true positive result. So again, AYN is at the top, ADE and AYE are in the middle, and MPA is at the bottom. And this time you can see that in addition to the stable isotope, AYN and both its qualifier ions are present, AYE and its qualifier ion are present, and at the bottom you can see that we've got the MPA and both its qualifier ions. So this is a barn door positive, and obviously it's also a delta variant. So this next slide is just summarising the concentration of the AYN peptide for samples that were PCR negative and PCR positive. The PCR positive samples have been split into those with a takeoff value of greater than 20, so that, in other words, the low positives, and those with a takeoff value of less than 20, which obviously are the more straightforward positives. And what this slide is really showing you that there is no really clear discrimination between a positive and a negative in terms of absolute concentration. And again, this is the same information, but for the other two peptides, AYE and MPA. And again, you can see that there isn't absolute discrimination between a positive and a negative. We've also been looking at the data to see if we can extract any other information out of it. So had a look at whether how the AYN correlates with the takeoff value. And you can see that there's broad correlation, as you would expect, but not particularly good correlation. I think the one that correlates the best is actually AYE. It's got an R-squared value of 0.779 though, so it's not really brilliant. Also been looking at ratios. This box whisker plot just shows the ratio of the AYN to the other two peptides. And we were looking in case it added some discriminatory value. But again, you can see for both peptides, the range is actually quite large. So we'll continue to go through the data and see if we can draw anything else out from it. And obviously, I think it's probably worth mentioning that at some point in the next couple of weeks, I'll be sending all this data onto Waters as well to see if Hans can do anything more sophisticated with it. In terms of the sensitivity and specificity, this is probably the most interesting bit. Of the 325 swabs we managed to collect from the St Thomas's Emergency Department, we had to exclude 10 because there was either insufficient sample volume or there was an inconclusive PCR result. And that left 315 in the final comparison. And we found that the mass spec method had a sensitivity of 75% and a specificity of 100% compared with PCR. Again, Cohen's kappa was greater than 0.83, which is consistent with excellent agreement between the methods. We also looked at those samples with a takeoff of less than 20 as a separate group, and you can see that that increased the sensitivities quite significantly to 92.9%. So I think in conclusion, the Waters SARS-CoV-2 kit has been validated on VTM patient samples. It's got acceptable analytical performance, and we also found the method to be very specific. It's not going to give a false positive, which is a definite advantage. And I think in terms of the sensitivity, it performs well at the lower takeoff values. And although at the higher takeoff values, its performance isn't as great, it's not actually, it's comparable to other antigen tests that are available and used routinely, such as the Roche Alexis assay. So I think what we've also shown is that if the process is automated, it could be suitable for a routine clinical environment. But I think for me, what this is really doing is kind of demonstrating the art of the possible. I think the Moonshot project has demonstrated what can be achieved and just how quickly if industry, academia and the NHS work together in partnership. And I think that's really important. And I think it's also highlighted the benefits of investing in high-end kit in clinical laboratories. But again, for me, I think the real take-home message is that clinical labs can do peptide analysis. We've got the kit and we've got a workflow. And for me, that means we have a whole new set of biomarkers available to us. And the question really is just which one we want to start with. So obviously, Moonshot has been a massive collaborative project, and there are loads of people that have been involved and need to be acknowledged. I obviously haven't captured them all on this slide, but I very much hope I've got the key players. So there's the rest of my team in Viapath, in particular Kate John and Kaylee Davis, who have been helping on the lab side, and Penny Cliff, who is my contact and very friendly virologist. And then Kevin Mills and his team from the Institute of Child Health, they were our partner P1 laboratory, and they were fabulous to work with. And obviously then there's the team from Waters, and then those people that were working directly for the Department of Health. I'll stop there, and if anybody would like to ask any questions, I'll be more than happy to try to answer them. Thank you.
So there was a question about the take-home messages in terms of the peptide assays, and I think for us it was the need to include multiple target peptides during the validation process, and also particularly for the COVID, we didn't have a marker of swab integrity in there, which was unfortunate because our results would perhaps have been a little bit more easy to interpret, but because we were using a ciscapper, that actually prevented us from going back and adding in a potential marker in real time. So I think that's something to bear in mind. So there is massive advantages of the ciscapper in terms of sensitivity, but it does mean you can't just add additional things into your assay, which is fine. But I think the lead time for ciscapper to produce new antibodies is between 6 and 9 months. So it's just something to bear in mind. I'm just going to have a look and see if there's been any more questions. No, there's just one other question that I've got in the chat. About what else we learned during the assay. So we're actually in the process of doing a formal lessons learned review, which is something we're quite keen on in England. Sorry about the background noise. So we've just contacted everyone who's involved in Moonshot with a questionnaire asking their thoughts on what went well, how we could improve things in the future, so that if we ever do another collaborative project in this way, we will be able to focus in on what worked well and avoid some of the areas where we perhaps made mistakes. Which I think is important because there were so many people involved, it was difficult to keep track of where everybody was at some point in time. Just checking the chat, I don't think there's any more questions. So I guess I'll hand back to Aaron.
Thank you, Dr. Carling, for that informative presentation. We will now start the live Q&A portion of our webinar. And if you have any questions you want to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have some great questions coming in. Our first question, what did you learn from the Moonshot project?
That's a good question. I learned a lot. I think I learned about peptide analysis, which I found really useful and I'm quite excited about. And I believe that in the States, you do a lot more with peptides already in the routine clinical laboratory than we do. So I think that that's an area that we would like to really expand, not necessarily expand into, but start looking at in more detail. And I'd actually be keen to hear from anyone who's got more experience than we do with peptide analysis. But I think also I learned a lot about working as part of a team because there was obviously quite a lot of people involved in Moonshot. Everybody had an individual role, but we also needed to work together. And on top of that, we were kind of geographically dispersed across the whole of the UK. So that was quite challenging. So yeah, it was definitely a steep learning curve, but it was a very enjoyable experience.
Thank you so much. And Dr. Carling, will the method be able to detect the new variants?
So that's a very good question. So obviously it depends on what the new variant is. So until the Delta variant came along, the method wasn't affected by any of the mutations that we were aware of. So for example, the Kent variant and the California, the South African and Brazilian, I think all of those mutations were predominantly on the spike protein, so they didn't affect our NCAP peptides. So in the event that we get a variant like the Delta variant, which does affect the NCAP peptide, the first question is whether it affects one of our target peptides. And then as with the Delta variant, it's about whereabouts on the target peptide it is and how that affects the binding to the ciscapper. So we were lucky really with the Delta in the sense that although it did affect the ABE peptide, the ciscapper the affinity was only decreased by about 20%. So we still captured the peptide successfully. And I guess that's the beauty of mass spec, because just by updating our MRMs to reflect the change in the peptide, we were able to still detect the Delta variant. And because obviously we have got 3 peptides in the method, I guess it would be not impossible, but unfortunate if a variant came along which had mutations which affected all 3 peptides at the same time. So I guess, worst case scenario, you may lose one, but hopefully the presence of the other two will still allow you to detect the COVID.
Thank you so much. And I want to thank our audience for these great questions. Our next question, why did you compare to the OS Diagnostics Method when validating the kit and not the Takehome assay?
Yeah, so that was the way the project worked. So in the first part of the project, as part of the Moonshot, we'd already got an agreement to do the comparison with the Crick laboratory in London, and they were using the Takehome assay. So when we went on to validate the Waters kit, that was just done in my centre, which is St Thomas's Hospital in London. So we had to validate the PCR assay, and we had to validate against our own PCR assay, and that our virology department were using Oz Diagnostics. I was a bit perturbed at first because the Oz Diagnostics assay measures things in terms of takeoff value, not CT. So that took a little bit of getting our head round. But I think in terms of the practicalities and the validity of the validation, it doesn't really make any difference because you can't compare, my understanding is anyway, that you can't necessarily compare the CT values from one PCR assay to another anyway. So yeah, it's just the way that things worked out for us.
Thank you so much. And we have one more question coming in. Can you explain the criteria used to classify results as either positive or negative?
Yeah, so this was probably one of the most interesting aspects of the project, because one of our jobs as the P2 laboratory was to work out exactly how to do that. So we were measuring the 3 target peptides, and for each peptide we had a quantifier ion and 2 qualifier ions. So we would look individually, just visually, at each peptide to check that the quantifier and the qualifiers were there. And if they were, we would class that as positive for that individual peptide. But then we would look collectively at the 3 peptides, and we would want to have 2 out of the 3 present before we would classify the sample as positive. And then we were lucky because, as I mentioned earlier, we were able to send the data on to Hans Vissers at Water, who looked at it retrospectively and used much more sophisticated techniques than me sat in front of the mass spec looking at the screen. And he actually came back and validated what we'd found using sort of more sophisticated algorithms. So yeah, so that was good. And again, for me, that was one of the learning points of the project, because certainly in the UK, the way we process our data post-analytically is often quite basic. And I think that there's things that we can take forward and learn here and apply to some of our other assays. My day job is metabolic biochemistry, so I think I'd like to see how this would look with acyl carnitines and amino acids, things like that as well.
Dr. Carling, this has been a pleasure. Thank you for your presentation today. Do you have any closing remarks you'd like to provide for our audience before we go?
No, just thank you very much for inviting me to speak to you today. Hope you found the talk interesting, and if anybody does have experience of peptide analysis in the clinical laboratory, I'd really like to hear from you and hear about how you've got on and what analytes you've been measuring. That'd be really useful for me.
Well, thank you again, Dr. Carling, and thank you for your time and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Waters Corporation, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years till November 2023. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Stay healthy. Bye-bye.
Bye.